So this is the Conrad Prebys Center for Chemical Genomics. The compound was discovered right here in this very room. This was actually done here using microscopy-based assay, which means you're actually evaluating visually a cell and looking for the impact of a chemical on that cell by a visual cue. We screened, in this case, 700,000 chemicals and found that amongst those, just 2 that actually were able to elicit this response in a cellular assay that we were after. And that was the very beginning of SBI 810. SBI 810 is the result of nearly 10 years of research. There's been a collaboration between the National Institute of Drug Abuse, which has been looking for completely new treatments with new molecular mechanisms of action to be able to treat addiction. When you alter it in the brain of an animal, has profound anti-addictive properties. What we found with our new compound, 810, and its other compounds in this class, they have a very different way of binding to the receptor in a different place, and because of that, we call these biased molecules. They are able to bias how they signal and therefore give us all of the positive sides without the negatives. And those side effects include profound hypothermia. We actually now know exactly the pose, how it binds, where it binds, due to a cryo-electron microscopy structure in which we actually have a snapshot of the molecular mechanism and how it works. So that's super exciting. The compound itself is gone through many tests and many animal tests showing activity in multiple models, not just of opioid use, but methamphetamine and cocaine, where it is shown to reduce the addictive properties of those molecules. So this SBI 810 is now a clinical candidate, and from declaring something a clinical candidate to being able to go to first-in-human dosing, there is a set of rigorous testing that needs to be conducted in a regulatory way. So you never know what potholes there are to fall in this process, but a realistic time frame to conduct those preclinical studies is about 18 months. You know, even with a bit of buffer, so let's say 2 years, something in that time frame before we get to the phase 1 clinical trial. And that phase 1 trial is all about safety, not efficacy, and that is, again, a mandated process by the FDA.